Turn it on camera.
Welcome to today's Psychiatry Grand Rounds. We have with us today Ingo Wilhun, who's PhD, who's now with our Department of Psychiatry and Behavioral Sciences, but he may be moving to Amsterdam or Eastern Washington, and we'll know that in the very near future.
It has been and it is still to the current date. Like these graphs show you here that about 200 million people on an annual basis abuse illicit drugs, and that makes about 5% of the overall world population. Interestingly, only about 10% of these 5% are the ones that are considered problem users, and I'm going to get back to that once we move on with the talk. Of course, these numbers differ in terms of what drugs we're looking at, if it's alcohol, nicotine, or illicit drugs. And the age range that's most affected are young adults. Since this differs in terms of what drug we're looking at, like the drug that I'll be talking about the most today is cocaine. And with many developments in the world, like the US also here is one of the front runners. We do have over 1% of prevalence in the abuse of cocaine in this country. And as probably some of you are aware, is that if you think that is bad, it was a lot worse actually in the 80s when taking cocaine was more like smoking a cigarette. It has decreased since, but it has been very stable, and there's no indication that this is going to change anytime soon. So I told you there's problem users amongst the users that recreationally try cocaine. And the difference really between these recreational experimental users and the actual addicts is that there's a progression that takes place over time. And with time, the intake becomes more habitual and in the end becomes compulsive. And these are the criteria that you find in the Diagnostic Statistical Manual that are used to diagnose drug addiction. And besides the first two, get tolerance and withdrawal, pretty much all of these symptoms have to do with the description of the loss of control of their behavior. And I'm putting up some pictures here of celebrities that at least some of these faces should be familiar with you that had problems with drug abuse in the past. And the fact that there's such in the public view, we have some idea of what the addiction of what the phenotype of the affliction looks like. Meaning there is it starts with a period of stress or some kind of interest in the drug, and then people realize that the drug is not good for them and they stop taking the drug. But then there's relapse, and this revolving door keeps taking place in the future from there on. I also put these pictures up here because I think it points towards an issue that we have in society. Even though clinicians have more and more gone towards the idea that we're having to do with a disorder, like a behavioral disorder here, and not just a weak character or people that are just like to have too much fun. So there's a bit of lack of empathy still in society for this type of disorder, and it's very controversial. So I told you that there's a progression that takes place. The behavior changes over time with increasing drug intake. And we usually start out, some people are more vulnerable to trying the drug than others. Some people are more impulsive, which is one of the main traits that determines whether people will try the drug. And then during the initial phases of drug taking, we find that drug taking is still very much goal-directed, meaning the drug is taken for a specific purpose. Either it has it makes you feel very good, there's a high that's being chased, or it actually alleviates psychological symptoms that are present already. With time, this becomes less so the case. So the behavior becomes a habit, meaning it's outcome independent at this point. It doesn't even matter anymore if the drug makes you feel good or if it does have any effects that you're interested in. So it becomes outcome independent. And with further use, which usually is marked by an escalated intake of the drug, drug seeking becomes compulsive, meaning at this point, it's not even outcome independent. The drug is even taken in the face of negative consequences. So even if there is some kind of punishment or some kind of people lose control of their lives, they lose their jobs, lose their families, they're still taking the drug, so it becomes compulsive here. We know at this point that craving for the drug and the compulsion are influenced by learning or conditioning. And this conditioning is already present very early on, and something that I'm very interested in. And what happens, for example, is that stimuli that are associated with the drug intake or the availability of the drug become very powerful. They become conditioned stimuli. And they often have something to do with the resumption of drug taking after a period of abstinence. So during my time as a PhD student, I already had some studies that related to this. And what you see here is really two coronal brain sections of a rat brain. And these rat brains are stained, or what we're seeing here is the expression of c-fos. C-fos is an immediate early gene that is expressed in association with the neuronal activation. So whatever lights up here as yellow, as brighter, has shown us that there is more activation in this brain region. What you see outlined in white here is the striatum in this rat brain. And in the left picture here is a rat that's only been treated with cocaine, and you see how the striatum lights up in its activity. But if the cocaine is given during a procedural learning task, so like a learning task that's not related to the drug taking itself, if this is combined, you see a potentiated response. And this is specifically the case in the dorsolateral aspects of the striatum. And this is down here just like the differential c-fos expression. And you see which areas are significantly different between these two conditions. Interestingly, this really only happens during the learning phase of this task, not before or after the learning. So before and after the learning, you still have an effect of cocaine on this brain region, but it's not potentiated by learning or by the behavior in the task. And these dorsolateral brain regions here in this graph is what you can see is that the inputs that go to that part of the striatum are the ones that are coming from the sensory and the motor cortices in the brain. So we have an effect on the brain region that we know is very much involved in the establishment of habits. So besides the drug being rewarding and producing a high, it also affects how habit formation is taking place in the brain. So there's two independent effects. So the c-fos expression that I just showed you is driven by dopamine and by dopamine receptors in the striatum. However, even though we know that these strong effects exist in the dorsolateral striatum, the field has really focused on a different part of the striatum and associated the ventromedial striatum with what we consider as important in the development of drug addiction. And the reason why this ventromedial striatum, or the limbic part of the striatum, some people refer to it as the nucleus accumbens, is so popular is because in microdialysis studies where people have measured the release of dopamine in the striatum, we've found that pretty much any drug of abuse elicits the increase of dopamine in this brain region. So people started thinking that this might be the common pathway. Even though all these drugs have different effects in general on the brain, they all have the same denominator, which is dopamine release in this ventromedial striatum. And this has led to the dopamine hypothesis of drug addiction, meaning that this seems to be like the most important event in the brain that leads to the abuse. So with more sophisticated techniques, we have now found that it's not just these tonic or slow changes in dopamine concentration in this brain region that are affected, it's also the brief phasic release of dopamine. And this is important to us because these phasic signals in the brain are driven by drug-associated cues. So conditioned stimuli just by itself can increase, for a short period of time, the amount of dopamine in the striatum. So knowing all this, but still knowing that there is like this effect in the dorsolateral striatum, the question is, how can we integrate these two pieces of knowledge that we have? And what you see down here is the reason why we see difference between ventral and dorsolateral striatum cannot be due to the fact that there is more or less dopamine in one of these brain regions. This is another coronal brain section of a rat brain, and it's stained for dopamine. So you see that there is dopamine present in both to the same amount in ventral and dorsolateral striatum. So one idea that has been brought forward is that maybe these brain regions play different roles at different time points. So the dorsolateral striatum, or the sensory motor aspect of the striatum, is thought to be involved in like the later stages, which have to do with habit formation of potentially compulsion. I'm going to get back to that later. And in the initial stages, we think that the behavior is driven by the ventral striatum, by this limbic aspect. And I'm going to try to test this hypothesis in what I'm going to show you in the following slides. Briefly, like what what has brought me to the lab that I'm in and has brought me to the University of Washington in Seattle is really this squiggly line here. And what this is like a 20-second trace of dopamine concentration in the ventral striatum in response to the presentation of a drug-associated cue. And right before here, the animal, like the rat in this case, has responded to get the drug and the drug-associated cue. So we had an idea of what happens in the ventral striatum. What really drove my research was...
Say that again. Does that explain that graph real quick?
So what you see here is dopamine concentration in the ventral striatum over 20 seconds in the ventral striatum. And what you see here, the black arrow is a lever press of a rat. And right after the rat presses the lever, you see that the drug infusion goes in and the cue is presented. And that produces an increase in dopamine concentration. So I'm going to get back to these traces more later. What we don't know is what happens in the dorsolateral striatum in this kind of setting. And what we also don't know is what happens when abuse or use is extended for longer periods of time. What happens in these brain regions to these signals? Are they changed? Do they stay the same? And how does it affect the behavior? So lucky for us and not so fortunate for rats is that animals, especially rodents and other mammals, like the same drugs that humans do. So we can model a lot of these behaviors in rats, in my case. And there's a number of different animal models. We can test different aspects of drug abuse. But the gold standard remains drug self-administration because the active intake of the drug has different effects in the brain than the inactive administration of it. So it's important that the rat seeks the drug itself to study this behavior. About 15 years ago, maybe a little more, the discussion arose of we've been studying this drug use behavior for a while now, but are we really studying addiction, or are we studying sort of like the recreational aspect of drug use? So as I told you earlier, there's a lot of people that try drugs, but only about 20% of them actually become addicted. So do we really study this addicted population, or are we just studying the effect of drugs on the brain? And there have been different models that have been brought forward by different groups in the field. And one that I'm going to be using and I'm going to be presenting some data on is simply like what's called the extended access to cocaine paradigm, meaning there's two groups of rats. One gets access to the drug 1 hour per day for several weeks, and the other group gets access to the drug for 6 hours per day. And the 6-hour one is supposed to be the one that is addicted in the end. And what happens if you do that, just in terms of behavior, is what you see here. These are the days of drug intake. The more days you have, the more the intake escalates. So you have more and more drug. And that's also true for the first hour of those 6 hours. So it's not just the overall amount, it's like the rate that the animals take the drug with. And you see in the black circles here are the ones that have 1 hour access, so they have a stable intake over time. So this model is one of the main symptoms that we see in addicts as well. Now, the squiggly lines that I showed you earlier, the way we measure dopamine concentration in the brain is by using a technique that's called cyclic voltammetry. And we have a very nice time resolution with that. We apply a voltage to the electrode that sits in the brain implanted for about 8.5 milliseconds. And what we do during that time is we ramp up a voltage and then we decrease the voltage in this triangular waveform. And we capitalize on the electrochemical makeup of dopamine. So what happens if a certain potential is raised and decreased like that, is that dopamine is oxidized and reduced. And this reaction actually produces a current that we can measure on the same electrode that we applied the voltage with. So since this is like a time-sensitive effect, we can then plot the voltage that we applied against the current that we measured, and we get a chemical signature. And using a principal component analysis, we can then fish out of the currents that we measured, which ones are due to dopamine. So really it's like measuring the concentration of the molecules in the vicinity of the electrode that can be reduced and oxidized. And that then gives us like an output of how many molecules are in this brain region that we're looking at. Just briefly, important for us was since we started implanting these electrodes chronically, which has been a new thing when we started in the lab about 5-6 years ago. So far, people had only used this technique acutely, so they just lowered the electrode, could measure, but then could not measure again a week later or so. So for us, it was important to span this progression of drug intake, this progression of the behavior, and actually be able to like take different time points in sample. And this is just showing you that the electrode doesn't do any real damage in the brain, at least not on a large scale. We stained here for astrocytes and microglia, and for dopamine. And this is where the electrode set. Here's like the shaft of the electrode, and here's the sensing end, which is really what we measure. And what we find is that there's no accumulation of astroglia or microglia, which means there's like not much inflammation. And also we don't see a decrease in the concentration of dopamine around the sensing end of the electrode. There's some decrease on the shaft, as you can see here, which is like this point right there. But we don't decrease the amount of dopamine, and we do not increase inflammation or produce inflammation in this brain region. In terms of functionality, of course, since we implanted for the first time for these long periods, we wanted to see whether the electrode itself changes. Does the sensitivity change over time? So we implanted electrodes for different amounts of time, 1, 2, and 4 months here. And we see that the sensitivity of the electrode, if we take it out of the brain and then measure how much dopamine is can be measured with the electrode, that there's no change over time. And then like the critical test was then also delivering food pellets unexpectedly to an animal always or very reliably produces an increase in dopamine in the ventral striatum. So if you present a reward to the animal that hasn't been expected, you see an increase in dopamine. And that increase doesn't change over the period of time. So you can do that even 4 months after implantation, and the electrodes are still sensitive. So much for just validating our tool. And this is one of my clients in his full combat gear and in his natural habitat, which is the operant box. So what the animal is doing here right now is poking its nose into one of these receptacles here, and this receptacle is connected to a computer that then will produce an infusion of cocaine through this intravenous catheter. These are the catheters that are implanted into the jugular vein of the animal, and is then connected to a microinfusion pump. And as soon as the animal pokes its nose in the correct hole, it'll receive an infusion of cocaine through this catheter, pretty much straight into his heart. And at the same time, we're measuring these the release of dopamine in the brain, and this is just like the connector and the head stage that then transmits like the signal to the computer, so we can have like an online readout of what's going on. And this is just the infusion pump that then will send the cocaine solution through this tubing here into the back of the animal. There's also, so this is like just showing you that we measure over the course of 3 weeks in this first experiment, and we're using an FR1 schedule, which pretty much means that every single nose poke that the rat is attempting into the correct hole, there's an active and an inactive hole, just to like control for overall activity in the box. Every nose poke in this correct hole here leads to an infusion. So it's a fixed ratio 1 schedule. At the same time, we have electrodes in 2 brain regions to measure, and then this is like this is an important piece of to understand what I'm going to show you in the following slides. So what I'm going to show you is usually like 40-second traces during a period when the animal decides to nose poke. So like this is when the animal nose pokes, and there's a 20-second presentation of the conditioned stimulus, and also the infusion of the drug takes place in this period. And this 20-second period here is also a timeout period, meaning any other response the animal produces in this time period is not leading to any other consequences. So the animal is not able to take more drug in these 20 seconds. Only after that it can decide to take more. So the animal is free to take as much drug as it wants in this case in a 1 hour period, and it's up to the rat when it takes this drug. So which is similar to like the human condition where we can decide when we want to take the drug. So before I show you any more graphs, I'm just going to show you a video because I think it explains it a lot better. So this is a rat now with a top view of an operant chamber, and down here you'll see the dopamine concentration in the ventral striatum, and then you see below in red the dopamine concentration in the dorsolateral striatum. And this rat has already taken a few infusions of cocaine, so it's very active. And this is the active hole, and it's soon going to approach the active hole and poke its nose in there. And there you go. And when you see that there's a Q light coming on, hard to see, but there's a cue light coming on, and then there's a tone coming on that you can't hear right now. At the same time, the infusion goes in. So you see in the ventral striatum that there's a strong response, while in the dorsolateral striatum, there's not much going on at this point. So far, so good. And then if we look at the same rat 2 weeks later, so like 2 more weeks of access to the drug, if you look at, I kind of want to go to the next one. So this is the same rat, again in the middle of the session, has already taken some cocaine and is now approaching the active nose poke hole again. And I believe this is the same video I just showed you. Let's see if we can get the next video.
Click outside the video.
There's still another video I'd like to show you, though. Let's see. Does it seem familiar? Is it the same video or not? Money question. And it is not the same video. Good. Okay, so here, this is the same rat 2 weeks later. And at this point, now we have a response in both brain regions. So nothing has really changed except that we have more access to the drug over time. So if you average these nose pokes over the session and across animals, you see that this holds up. Like there's a strong response in the ventral striatum, while there's nothing in the dorsolateral striatum in the first week of training. But then if you look at the second week and third week, you see that in the ventral striatum the signal is present throughout, even though it decreases a little bit. While in the dorsolateral striatum the signal comes on in the second week and stays on in the third week. And we know that this signal that we're looking at there is not due to like their actual motor act, like the behavior itself to move towards the nose poke hole. Because if we look at compare it to responses to the inactive nose poke hole that doesn't give him the infusion, we don't see an increase in dopamine for that. And at the same time, if we deliver the conditioned stimulus that's associated with the drug intake or the drug delivery, that this stimulus on its own, if delivered unexpectedly to the rat, has the same effect as like what we see during the session when the animal actively seeks the drug. And what I'm not showing you here is that if you just give an infusion of cocaine, that is something that like the pharmacological effect of the drug itself is not coming on until like 10, 15 seconds later. So what we're really looking at is like it's something that's related to the drug stimulus itself, I mean the conditioned stimulus. So now, as interesting as that is, of course the question for us was, how does it affect behavior? What is its relevance to the rat's behavior? And what we know is that, as I told you earlier, if we give the animals 1 hour access per day, their behavior in terms of how much drug they take is very stable. So like it doesn't change. Like they pretty much take the same amount 3 weeks as in the first week. And what I showed you here is just like the development of the dorsolateral signal. So there's nothing the first week and then second and third week. So we figured, well, maybe like it doesn't steer how much drug the animal's taken. Maybe it's just the locus of where the rewarding properties of the drug are being encoded in the brain. So meaning that maybe in the beginning of training, the ventral striatum is really like what makes the drug rewarding, this effect there. And then later on, it's in the dorsolateral striatum where the rewarding properties are encoded. So if we give a dopamine antagonist infused straight into the dorsolateral striatum, what we see is that if we do that in the third week, we see an increase in drug intake, which is often like considered like a compensatory response. So the animal wants a certain amount of dopamine in response to taking the drug, and if it gets less because the receptors are blocked, it'll just take more drug to compensate for that. And so that will make sense to us because the signal is there in the third week, and this might be exactly what we thought it is. However, if we do the same thing, we infuse the antagonist in the first week, the same thing happens behaviorally, but we don't have a signal at this point yet. So this crushed our idea of like maybe just like a shift in the locus of where the reward is being encoded in the brain. So we remembered that we know that the dorsolateral striatum is very much important in action selection. And I told you earlier there's an inactive and an active hole, and also that there's a timeout period of 20 seconds where the rat's nose pokes are useless. It doesn't get anything for it. So if we plot these like non-successful responses in the inactive hole and the responses during the timeout on this graph, we see that in the first week, the animals still make a lot of what you could call mistakes. And then later on, their targeting of the active hole is much more straight. So we figured if we look at what happens in response to the dorsolateral striatal dopamine receptor blockade, we see that it doesn't do anything really to like these non-successful responses in the first week, as we would assume if there's no signal. But if we give this antagonist and block the receptors in the third week, when we have a signal, it reverts back to what happened in the first week. So like their goal-directedness is decreased. So this is usually this is an interesting part of my talk, because I usually try to look at my audience, how people react to this, because here's this German scientist that talks about the efficiency of drug intake, really. So out of all the things that come along with drug abuse, I'm focusing on how efficient drugs can be taken by these rats. In Cambridge, in the UK, this produced a lot of laughter, because they figured, oh great, now we found a German brain region. While at UC Irvine, for example, I didn't get a single smile for this. So the idea is really I don't think about it as much as efficiency, but it's more like a change from exploring this drug environment to exploiting it. And this might be mediated by the dorsolateral striatum. So more of a prioritizing this behavior towards the drug over any other behavior that the animal is capable of performing in this box. So this is a bit of switching gears now. These are not just colorful graphs, they also have a reason why I put them up, and I'm going to explain them just for a minute because it seems a little complicated. So again, on this axis here, we have those 40 seconds that have been shown you so far, and this is the line here shows where the animal performs the nose poke. We have two brain regions. This is the ventral and this is the dorsolateral striatum, and this is for the traces that we see here is for every single nose poke the animals do in this session. So there's 60 minutes here. And every trace that you see here is one approach behavior towards the nose poke receptor. And then this is the amount of dopamine released. So one thing I want to show you here is, first of all, there's a good amount of heterogeneity in what we're measuring. So it's not always the same throughout the session how much release is induced. And the other thing that's somewhat important here, and it's going to be my hook for the next slide, is that the dorsolateral striatum is really not responding much in the beginning of the session. So these first 10, 15 nose pokes here don't really produce anything. But it comes online during a time when the ventral striatum seems to be a little weaker in its response. So the question that posed itself from that for us was, is there any kind of relationship between these two brain regions and how they signal? How are they connected? And here it gets a little more complicated, but I'm going to try to keep it simple. And there's been a very interesting study by Suzanne Haber that looked into how ventral and dorsal striatum could potentially talk to each other. So a long-standing question in the field, this is a monkey brain, or a schematic derived from studies in a monkey brain. What you see here is the striatum in the monkey, and these are the ventral and limbic aspects, and here more dorsolateral aspects. So we have these loops that go from the cortex to the different parts of the striatum, and then go from there through the thalamus back to the cortex. And these loops go round and round and are activated consistently. But they can't really talk to each other from what we can understand so far, because they're isolated from each other. So Suzanne Haber suggested that they might be talking to each other through projections in the brain that have something to do with dopamine. And I'm going to jump from this to the rat version of the same idea. And what you can see here again is a coronal section, two hemispheres, striatum up here, and this is where the dopamine neurons reside in the midbrain. And the idea is that ventral striatal dopamine neurons, or dopamine neurons that project to the ventral striatum here, they have a projection, there's a reciprocal projection from the ventral striatum back to the midbrain. But this projection doesn't only go back to the neurons that it received dopamine from, but also to neurons that then will project to further dorsolateral parts of the striatum. So this loop would go back and forth until the signal goes from ventral to dorsolateral. It's this back and forth communication. And Barry Everett and his group in the University of Cambridge in England has done some very elegant experiments. I'm going to simplify them a lot. Basically what they've found is that, first of all, this behavior is driven by the ventral striatum seeking of the drug, and the drug cues is driven by the ventral striatum first, an idea that I introduced earlier, and then later it's driven by the dorsolateral striatum. What he showed is if the signal is prevented from progressing from ventral to the dorsal, these loops back and forth, if they're interrupted, or what they call a disconnection study, is that we don't see seeking if this serial connection is interrupted. And this is here just like a dose response to a dopamine antagonist that's given unilateral into the dorsolateral striatum. At the same time, the other hemisphere has a ventral striatal lesion. So like the ventral striatal lesion prevents the spiral from going into effect. So this study really then prompted like a study that we wanted to do with our results, or going back to our results. We figured, well, that is true for the behavior that this, if you disrupt the spiral, you don't see the behavior anymore later on. What if we disrupt the ventral striatum in our studies, we lesion the ventral striatum, and then measure dopamine release in the dorsolateral striatum on both sides of the brain? So if there's really a progression that goes on from ventral to dorsal, we shouldn't see a signal in the dorsolateral striatum here. And the neat thing about doing this study was really that if you lesion one hemisphere of the brain this way, the behavior stays intact. So they still take the same amount of drug. They still perform the behavior and seek the drug and take it. But one hemisphere stays intact. So we have a control region within animal. And this is what you see what happens in this side of on the healthy side of the brain. We have the same kind of like slow development of the dorsolateral signal. The second week it comes online and it stays online. And to our delight, if you look at the lesion side of the brain, if you measure in the dorsolateral striatum, this progression is interrupted. There seems to be still like a small signal that's even present from the very get-go, but it doesn't have this stronger, like this increase in dopamine signaling. So we figured whether this is really due to the spiral or not, we definitely have shown something here where there's a dependency of the dorsolateral signal on the ventral striatum. And this is not due because the dorsolateral striatum is not able to release dopamine anymore. Because if you look at what we call spontaneous events or events that are not associated with any like action in the actual task, both sides of the brain in the dorsolateral striatum are still able to release dopamine. So it's just like the specific behavior is not releasing dopamine anymore. And so this is we're coming to the last part now, which is really like everything I've showed you so far could be considered a giant control group for this experiment, because we set out to do this experiment, but we wanted to look into these addicted rats, meaning the ones that get 6 hours per day. So far I've only talked about animals that had 1 hour per day access. And this is just to validate that in our hands, like this paradigm works the same way as in anybody else's. If you switch the rats from 1 hour to 6 hour access, they'll escalate their drug intake. If you only look at the first hour, you see the same thing. They take more and more drug, while if they had short access, they would stay stable. This is just the same thing in bar graphs. So the question, of course, for us was, well, what happens with the signal that we've characterized so far in short access in this long access or more addicted animals? And what we see is that in the ventral striatum, this signal disappears over time. So we have the same kind of signal in the beginning, the first week, but the more of the 6 hour access the rats have, like the signal disappears, which is against some of the theories that are out there in the field regarding that the ventral signal may be the signal that encodes incentive salience, meaning that like the drug cues become more and more salient over time, and therefore the rat would take more drug because these cues are more salient. So this doesn't seem to be the case, at least in this particular task. The idea then was, well, if it's true that the dorsolateral striatum is taking over this behavior once it becomes more habitual, then we should see this. The ventral striatum sort of phases out, and the dorsolateral striatum takes over. But what we found is this. It really, the beginning of these measurements, it looks like it's the same as for the short access. There's no signal in the first week. It comes on in the second week, but in the third week it disappears. So our first impression was we were a little devastated because it seemed to suggest to us, well, it looks like dopamine has nothing to do with this. It's not there anymore. Which was, not just because I put so many hours into this, was hard to accept, also because I knew some things that have been studied in the field so extensively. And we know that rats, like humans, they're very rigorous in how they titrate their drug intake. So, for example, what you see here is this individual rats, and you program the pump differently so they get different doses per response. So if you give them a higher dose for each nose poke, they'll respond a lot less over an hour or 2 hour session, because they want a certain amount of drug, they don't want less, they don't want more, they want a specific amount. So they're titrating the amount of drug. And if you give a dopamine antagonist, as I told you earlier, if you give it systemically, for example, it's been shown 100 times that the animal will compensate this lack of signaling on the dopamine receptor by taking more drug. And this is really like, you can get like a really nice dose response with that. So it seems like dopamine must have something to do with the titrating. And this is even more specific if you, for example, infuse in the ventral striatum an antagonist, you get an increase in intake. If you give an agonist in the ventral striatum, you get a decrease, so they have already more dopamine signaling, they don't need as much drug. And then this study is just the pinnacle of this research, I would say, where this group around Roy Wise actually studied like in more detail. What you see here is like the concentration of dopamine measured with microdialysis, so on a time scale of like 100 minutes here. And you see that whenever the dopamine concentration drops to a certain level, that's when the animal takes the next infusion. Sorry. And so really, like, it seems like the animal is not even so much titrating its cocaine level in the blood. It's really titrating how much dopamine they have in the system. So after we've thought about this some more, we figured, well, maybe these drugs are escalating their intake in this paradigm because they get less dopamine for each of their responses. And unfortunately, we had like a fairly big sample, so we could start looking at into individual differences in our rats. And what we found is that in this sample that we took, there is each of these dots here represents one animal. Some of these animals in this test don't escalate their intake, and the majority of them does. So if we now start looking at our measurements separated by these two groups, we first looked into the dorsolateral striatum, and we see that these are the non-escalated animals, and these are the ones that escalate their intake. But there's no difference really between these different individuals in relation to their behavior. And that's true, there's no correlation between dopamine and their behavior. But if you look at the ventral striatum, you see this clear distinction of the animals that remain stable intake. They have a stable signal in the ventral striatum. The animals that escalate are really the ones that lose their signaling. So it reinforced our idea that there might be, the connection might be that they need to get more bang for their buck in a way, that they need to get, they're really like trying to like get more of the currency of this behavior, which in that case would be dopamine. And so this holds up even if you do a correlation. You find that there's an actual like a significant correlation at the third week between the amount of release and their behavior. And so, oh yeah, and this is like an important piece of information, as it just shows that between these two groups, the effect of cocaine itself doesn't change. So it's not like a pharmacological phenomenon where cocaine is just like less efficient in some of these animals. And it seems like that the animals that escalate also have a higher motivation to seek the drug. So it seems to be somewhat meaningful, like this, to make this separation. So the idea for us was then, what was next? What do you do in the clinics, like when you have a lack of dopamine? One way of targeting that is to give L-Dopa, which is like a precursor for dopamine, so we could increase the production of dopamine and see what happens to this behavior that we've looked at so far. And we know that this phasic signaling that we're measuring can be affected by L-Dopa, which is not a surprise. Like you increase phasic signals if you give L-Dopa. And you can have this effect if you give L-Dopa for several hours, which is important for us because we gave these animals 6-hour access to the drug itself. So it's actually suitable to use it in our paradigm. And so this is really the result of that. What you see here in orange is like the short access group, the ones that have 1 hour access per day. And in purple, you have animals that have 6 hours, and see how they escalate. And then we asked, well, can we prevent this escalation by giving L-Dopa? So like starting at this time point here, we started giving a systemic infusion of L-Dopa half an hour before the session started in each of those days, and that's what these open purple circles represent. And you see how the animals that received this L-Dopa injection and had 6-hour access, they did not escalate their intake as much, and they stayed at the level where the animals that took only 1 hour per day. Clinically more relevant then seemed to us, well, we're probably not going to see, we're not going to be able to give everybody L-Dopa to prevent them from escalating their drug intake. So what if a patient presents itself and has like this sort of like really like escalated intake, is this anything that could be useful in the clinics potentially? And so we let the animals escalate, and then we gave them L-Dopa later, and we see that we can reverse this intake behavior. So really, I mean, we don't have the illusion that this is going to cure drug addiction, but the thought that we're having at this, which might be promising, is that maybe this could do something like along the lines of harm reduction. If you have a patient that has this escalated intake, can you bring him down to like what a recreational intake person would do, and then build strategies on top of that, like have the patient more responsive to what you're trying to do. And there's been like two very rather small clinical trials that actually found like if you use a token-based like abstinence program where the person is rewarded for being abstinent, if you also give L-Dopa, you actually get a better outcome. But they're really small studies, and hopefully there'll be more in that regard in the future. So in summary, what I've tried to tell you is that phasic dopamine release, first of all, is region-specific within the striatum, which is not trivial because there have been suggestions, like in the field, like and like a very influential theory proposed that there is one dopamine signal that goes out to every brain region, and it's the same everywhere. So in our hands, this doesn't seem to be the case. Dopamine signaling changes with the progression of drug use. So in the ventral striatum, we see the signal that's present early on, and it seems to have something to do with reinforcement. While the dorsolateral signal comes on later, and it seems to have something to do with the switch from exploration to exploitation, or the prioritization of this behavior. But it doesn't seem to have something to do with compulsive drug use, as far as you can see, because we see the signal come on in a group of animals that are definitely not compulsive yet. So it's not a one-to-one relation to that. And then importantly, we found that not as like had been proposed and was thought that the dorsolateral striatum was like the driver in this escalation of intake, we found that the ventral striatum seems to be the important player here, meaning that if there is stable intake, we find stable release in the ventral striatum, but if there is an escalation, we find this decrease, and this seems to be fixable with L-Dopa, at least in the short term. And the other thing I was trying to tell you was that there is potentially this spiraling connection, but at least there's definitely a dependency between ventral and dorsal striatum. So this dorsolateral signal that seems to have something to do with the automatic intake or like the habitual intake is dependent on what happens in the ventral striatum. So this progression is not just seen in behavior, but it's also like there's somewhat reflected in the progression in brain regions that are involved in this behavior. So going back to like our initial slide, what you see is that in animals as with humans, you have less, and you have this cycle of intake, and the behavior changes over time. There's more people that start out, and then there's like a subpopulation that moves on to the dysregulated or escalated intake, and this seems to be more related to the dorsolateral striatum. This surprisingly seems to be more related to the ventral striatum, but we haven't really addressed compulsive drug seeking at this point. So there's only a fraction of people that really move on to the next stage, and there's some data that I have now that I would like to pursue in the future that maybe give us some clues on what happens here in terms of in the world of striatal dopamine. So what I found is that that animals that seem to be more impulsive than others, and this is they've shown it by what you see here is like the responses during the timeout period, is 20 seconds after they got an infusion, where like no responses lead to any consequences. We find that escalated as well as non-escalated animals here in blue, they don't really respond more in this timeout because they know there's nothing that comes from it. But there's a subpopulation of the animals that are escalated that will like do more and more and more of these timeout responses that are like really like it's not clear at this point. Like that's something that needs to be explored. Is this an impulsive or a compulsive behavior potentially? And interestingly, if you look at these rats in more detail, you find that they have even a higher motivation to seek the drug than the non-escalated animals, and they also have a little bit more intake. But what's interesting to us is that in terms of what happens during this period of long access, 6-hour access to the drug, there's no difference really in dopamine release between these two subgroups. But these animals that become impulsive or impulsive-like later, they really have like a very strong dopamine response to the first exposures to the drug. So it seems like this might be predictive of what happens later on. So this is something like one of the routes I would like to go down to in the future. And then the other question is really, so I've showed you this progression of dorsolateral dopamine signaling, but we don't really know what part of this is really pathological. So it could be if the animals were responding for food, that we have the same kind of development where a signal comes on in the second week. So I developed a task where they will take the same number of reinforcers, whether it's drug or food, and then we want to look at what happens to the signal development in the dorsolateral striatum, so we can tease out which part of this might be aberrant, might be like what's producing actually addictive behavior. And then you could also try to see at what point in here will the animals prefer the food, prefer the drug over the food, and has something to do with the signaling changing. And lastly, I'm going to use some this very popular technique these days, the optogenetic manipulation of striatal dopamine release, where you just give a virus that contains a channel that is sensitive to light, so you can implant this channel and activate specific populations of neurons by shining a laser on those neurons specifically. And we have already expressed these channels successfully in the VTA, in the part of the brain where dopamine projections go to the ventral striatum. So what we're hoping to do is to shine the laser on the terminals, so we can really specifically activate dopamine release in the ventral striatum only and nothing else, and then measure what happens in the dorsolateral striatum to see whether we can manipulate and see what the dynamics are. Is it a one-to-one connection, or is it something that develops over time if you really stimulate strongly, and so on? And we've already successfully been able to elicit dopamine release this way. So the tracks are laid, but the studies have to be done at this point. I would like to thank the lab, and actually notice that one of my lab members actually made it here. So even the retreat is going on, and my colleague Vince Martinez made it here. So I'm very thankful for that. And Paul is the PI who's been very supportive and has really provided us with everything we need to do successful studies. Our collaborator Barry Everett in Cambridge, these are all students and postdocs, and some of them have already moved on. Actually, these two have graduated at this point. Thank you for your attention.
So in your dose escalation model, have you looked at other agents besides L-Dopa, specifically bupropion that's used to treat nicotine dependence, and some people think help folks with meth dependence and that sort of thing. And are you interested in doing that? How novel is this method for screening potential clinical efficacy of various agents?
So the first part of the question is, have we tried other agents besides L-Dopa to see what happens in this escalation model? So we have not yet. Cocaine is one of the the go-to drugs for studying drug addiction, but obviously it has different pharmacological profile than other drugs have. So it would be really important to study, first of all, other drugs of abuse too, and to study other agents that will influence the same brain mechanisms as we looked at so far. This is really only a brief first glance at what might be possible and what not. Obviously, if this holds up and is really representative for addictive behavior, we should be seeing similar effects with other drugs of abuse, since if this is representative for what is the common pathway for drugs of abuse. And also, if this is really the mechanism that is important, we should be able to use other agents to manipulate it. And the second part of the question was whether these type of studies, if I understand correctly, lead to translation into the clinical world frequently. To start out with, I think that in terms of psychological disorders, we're fairly lucky in when it comes to rodent research, because drug addiction is something that's you can model fairly well in the rodent, and there have been other studies in the past that have led from this basic type of research to clinical trials in agents that are actually now used to treat addicts. So there have been studies in Peter Kalivas' lab at MUSC in South Carolina, where he really found changes in glutamate transmission in certain brain regions that he thinks are important, and he developed or he used a drug that can actually change overall glutamate concentration by changing the function of the glutamate exchanger on glia cells. And so there are examples, but I don't think it's something that happens enough. Obviously, there's definitely a need for trying to push this more into the clinical world than we do so far.
I wonder if you're getting at something larger than drug dependence, but what we call immune reward dependence, which is compulsive. There's a lot of data.
Yeah.
Future experience we have.
So the question is whether this, what we're looking at so far, in terms of impulsive behavior and also like this whole model, is getting at something that's like larger than just drug addiction itself, whether it's more of like a general mechanism in the brain. I would very much agree with that, because I mean I don't think that we have specific brain centers that are really there to make individuals addicted or not. I mean these are behavioral traits that are present in individuals, and impulsivity is one of them. We already know that impulsivity, like people that are more impulsive than others, are more prone to develop addiction. So what we're really trying to do, I think, is to understand the behavioral traits that are then involved in addiction, or then dysregulated, or lead to dysregulation of other behaviors that are downstream. So I would definitely not separate this and say that this is something that is really only specific to drug abuse or dependence. I would think that there are behavioral traits that will also important for other impulse control disorders, for example.
Thank you so much.
Thank you. Thanks.